Hello, everyone. Thanks for joining us today. My name is Ed Creedon. I'm a senior project manager with AST Environmental. Our presentation today is going to be given by Edward Winter. He's Vice President of Remediation Products, Inc. And Ed tells me that his talk is going to run about 30 minutes or so, but I'd like to be sure you know that we will stay on as long as necessary to address any questions that you may have. So I hope you all take advantage of this opportunity to dig in with us today on this very interesting topic. I'm guessing that most folks are well familiar with this platform, but just quickly I'll point out there is a question tab in your webinar control panel. So feel free to enter your questions as we go, and we'll get into the Q&A once Ed concludes his presentation. And before I hand it off to Ed, I'd like to just take a few moments to talk about the RPI group. The RPI group is made up of Remediation Products, Inc., RPI. RPI is a developer of the activated carbon-based technologies known as BOS 100, BOS 200 and 200 plus, and CAT 100. Also in the RPI group is the company that I work for, AST Environmental. And the third part of the RPI group is the network of authorized installers. Now, RPI is based out of the Denver, Colorado area. AST is headquartered near Lexington, Kentucky and Midway, Kentucky, and I'm actually in Kansas City, and we have several other staff working remotely around the country. And there are numerous authorized installers located around the US, in Canada, Australia, and in Scandinavia. AST is tasked with national and international project assessment and design, as well as quality control of field operations, and we're also the worldwide distributor of all of the RPI products. And the authorized installers, this is a group of companies that have completed extensive training for field installation, and the many years of shared experience and acquired knowledge of this group is really a critical component of the overall success of our projects, because as I'm sure most of us here today know, the best designs are really only effective when the products are properly installed to make contact with the targeted contaminant mass. If you have interest in learning more about the field methods, how we apply the RPI technologies to different contaminant sources, how we place these products in a wide variety of geologic environments, I invite you to visit our website at astenviro.com, and there you'll find an extensive library of recorded webinars that go really well with that first cup of coffee in the morning. At least I enjoyed doing that kind of thing. So I hope you check those out, and you'll find detailed information on the RPI website regarding the various technologies and the science behind them. And that website, it's easy to remember. The name essentially describes the remedial process. Just go to trapandtreat.com, and you'll find loads of additional info there, including a listing of the authorized installers of the RPI group. Okay, so let's get to the fun stuff and let you hear from the Ed that you wanted to hear from today. I think you're going to find his presentation to be quite fascinating. He's got some amazing imagery. He's got a lot of well-researched information to share with us regarding the impact of particle size on activated carbon-based amendments. I will be back at the end of Ed's talk to help facilitate the Q&A session. So Ed, please take it away.
Okay, well, I'd like to thank everybody for attending. And this presentation developed out of observations I made at looking at soil cores. So these are not laboratory samples. These are samples that were taken from sites and examined. And I made an observation that I thought was worthy of at least further consideration, and that is the impact of the particle size on activated carbon-based amendments. Okay, so let's start with some key concepts. To understand the value of activated carbon, most of you already have a grip on the idea that it traps, that activated carbon can sorb contaminants. But the treatment aspect is incredibly important. You know, if you have trap but no treat, what do you have? You have a hazardous waste landfill, right? So to function as treatment, you need it to function as microbial habitat. And activated carbon must have sufficient surface features to allow bacteria to adhere to the activated carbon if it's going to act as habitat. Biodegradation on activated carbon depends on the biomass, how many microbes there are, the growth rate, how active they are, and the community structure, basically who's there. Activated carbon itself increases microbial community diversity and changes the microbial community structure. And these are the kinds of things we want to address today. So yes, activated carbon traps, but it is the microbes that treat. Before we get started, I want to orient you to bright field imaging. Now, this is the kind of microscope maybe you had in high school or college where the light comes in from behind the object that you're looking at. That's called bright field. And it has several issues, that you can work through, but you should be aware of. First of all, it has a very narrow depth of field. So when you're looking at something that's in focus, there's always lots of things around that object that are out of focus. The other thing that happens is you have the bending or diffraction of light rays around objects. And that bending also causes dispersion, the light to separate into its wavelengths. So if you look in the middle of the picture here, which I'm pointing at with my arrow, you'll see a red circle. And you'll see a black object with kind of a burgundy ring around part of it. All that is diffraction and dispersion. It's everything in the picture, which you're looking at right now, is black. If it's lighter, it's because it's out of the depth of field. If it's black, it's because it's closer into the depth of field. In fact, this is an image of colloidal carbon. Most of these smaller pieces, single pieces of colloidal carbon, will be about 2 micrometers in size. Some of the larger pieces are either an actual larger piece or they're a consolidation of multiple smaller pieces. This is an image of bacteria mixed with colloidal carbon. Now you'll see a lot of the colloidal carbon agglomerated in this picture. I'm not going to blame the product for that. That was me trying to get them all mixed together. But what you can conclude from looking at this is that the bacteria themselves are in about the same size range as the colloidal carbon. So on the right side, you'll see I have a picture. It looks like a black spot with green Mickey Mouse ears or something. The black is a piece of colloidal carbon. The 2 green Mickey Mouse ears are bacteria. Below the picture on the right, I have another one where there's a bacteria, maybe a second one, on top of a piece of colloidal carbon. The only thing I'm trying to demonstrate from this picture is that the microbes, which are green, are roughly the same size as colloidal carbon, which is black. And also note that a lot of the colloidal in this particular picture is kind of agglutinated, kind of mounded together. This is another picture very similar to the previous. These are microbes mixed with colloidal carbon, showing that the microbes are in the same range. In this particular case, this microbe is not a bacteria. This is probably a fungi. So the fungi are quite a bit bigger, but this is a very small fungi. Nevertheless, gives you a sense of size. So why is it important that microbes are the same size as colloidal carbon? Well, I like the picture that I'm showing you here on the right as perspective. This is 2 pieces of what we call GAC, granular activated carbon, that are kind of stuck together by biofilm. This is probably a combination of what's called extrapolymeric substance and microbes all woven together. And there are several things that you can see in this picture. The weaving, which I'm moving back and forth my mouse pointer. They built up biofilm on the edge of the carbon. At least that's the way it looks right here because of the the limitations of the microscope, and you'll see a little of it here. So why is it important? Because activated carbon itself acts as a habitat for microbes. So this is a scanning electron microscope image. If you look at the image on the right and the top, which I've labeled A, you will see what I call a large feature in the activated carbon. Sometimes these things are called macropores. The problem with using the term macropore is a macropore is anything over 50 nanometers. And there are tests for micropores, and the tests for micropores don't distinguish between a 50 nanometer micropore and something the size of the picture I'm showing you in A. So we will just stick with surface features. This surface feature is space. If you look in the picture below in B, there's a surface feature that's filled with bacteria. In this particular case, the bacteria are kind of linear as opposed to the round ones I showed you. They look more worm-like. But you can see immediately that if you are trying to provide space, a 2-micron piece of carbon would not provide you any space in which microbes can live. So the physical structure of the activated carbon itself is a place where microbes can find protection, where they can attach, and where they can build multi-species biofilm. Now, this should not be confused with the adsorption to the micropores, which is a key feature of activated carbon, and it's what you hear about most often. It is the trap. But right now, we're worried about how do the microbes live on the carbon so that you develop the treat. So to function as microbial habitat, activated carbon must have sufficient surface features, such as the ones I'm showing you in A and B, to allow bacteria to adhere to the carbon, to find that safe space to live, to build and grow biofilm. Now, this is kind of an example. Here are microbes growing on activated carbon, which is the top picture, which in this case is marked C. I've marked some of the microbes in blue. Now, clearly that is me putting blue on top of the microbe. That's not in the original picture, but that's designed to help you notice that all the linear tubular items that you're seeing in that top picture are bacteria growing on powdered activated carbon. If you compare it to the picture below, which is marked F in this case, you'll see that there are far fewer bacteria growing on clay. So it's been published multiple times that microbial growth on activated carbon is superior to most other materials, such as zeolite and sand as an example. Now, colloidal activated carbon, having an average of a 2 micrometer grain size, is actually in the same size range as clay itself, which is a little bit less than 1 micrometer to a little bit less than 4 micrometers, or a 1 to 4 micrometer range. Thus, I would proffer that it's unlikely that colloidal carbon would be a vastly superior habitat for microbial growth than the clay, which you see in figure F. Now, this is in contrast. This is bacteria growing thick on GAC, granular activated carbon, and PAC, powdered activated carbon. And these carbon granules were scratched from a site core. So this is not a laboratory experiment. This is a core that was pulled from the ground. So if you look at the upper one, the activated carbon with biofilm, you will see one grain here, which I'm circling now with the mouse. That's about the size of PAC. So that would be about 20 micrometers. This, which I'm now circling, the larger piece, is about the size of GAC, which is around 200 micrometers or so. And notice the biofilm along the surface. Now there's biofilm on top too, but you wouldn't be able to see that due to the character of the microscope. Notice the biofilm here, thick and heavy. Now I will show also that, as I scratched at that core log, I also pulled off some just straight biofilm, which I'm circling now. These are microbial cells. If they have any carbon content in them, I'm not really sure. Sometimes when they're so thick, you just can't see through them and you might interpret it as carbon. The larger pieces, I can know whether it's carbon. That's easy. So microbes build, not only do they grow thick, but they build structure. And what I'm showing you in this picture, these are two pieces of activated carbon, smaller range GAC, and you can see it looks like maybe there's something in between them. And actually, they are attached to one another by extracellular matrix, extrapolymeric substance, and piles of microbes. In fact, if you move over to the right side, you can see, right now I'm pointing at it with my mouse, there's almost like a tree-like structures growing out or off the surface of this piece of GAC. So this GAC is actually acting as a source, perhaps of nutrition, of energy, or just a surface to bind to for the growing of these microbes. There's a little tree structure there to point it out, the arrow I just popped up. Now there's also this. You can barely see it, but I'm gonna get that back down. Okay, there we go. But I have noticed that there's a lot of kind of odd little pointy floppy structures like I'm circling now, and that'll come up here in a minute. Okay, so biodegradation on activated carbon depends on biomass, which I've shown you, heavy biomass on GAC and PAC, growth rate, which we're not dealing with at the moment, and the microbial community structure. You're seeing in this slide actual structure being formed by the microbes. Now these two pictures are quite interesting, and I'm trying to show you the kind of patterns that I'm seeing. So on the left, you have a really thick, tight biofilm. The black is activated carbon. The root beer color is a heavy, heavy biofilm. Again, that's going to be made up of extrapolymeric substance and lots of different microbes piled into the biofilm. There's also those, again, tree-like structures. Isn't that really the most interesting picture on the right? I was so excited when I saw it, actually. But anyway, I call it expansionary. I have no idea what intent the microbes have, but it's this branching pattern as if they were reaching out into the larger matrix to find something. I just gave them intent, they don't think anything. Here's some other pictures, and this goes back to what I pointed out earlier. I've noticed that the bacteria tend to collect sometimes in these kind of almost pointy structures. On the left, it looks like that the bacteria built up so heavy they almost fell over. That's the way it looks. That's not necessarily true, but that helps you understand earlier why I pointed out on the right the seeming images that are hard to see as pointy structures, also heavy biofilm. So the assumption is that microbes will also grow on colloidal activated carbon the way they do on PAC and GAC. I have two pictures here. The one on the left looks like a kind of valley with fog around it, and the fog is really just being outside or taller than the focal length of the microscope. The black spots that appear to be lined up, and that's only appearance, are carbon the size of colloidal carbon. And I wish I could get them bigger and closer, but this is about the best I can do with something that is 2 micrometers small. And the best I can tell, especially if you look on the right, the smaller pieces, the bigger one is almost headed towards being the size of powdered activated carbon. I don't see any microbial growth. There might be microbial growth there. It's possible that I just can't see it. But if it is not near as extensive or interesting as what I've already shown you. Now, when you get a activated carbon product, all the products are a mix of sizes because of the way they're made. So even if you get powder, you will have some colloidal in it. If you get colloidal, you'll have some that are the size of powder, and then GAC can have all of it. So it's really primarily the major size in which the carbons are characterized by. But nevertheless, looking in these pictures, you don't really see any growth. Now this is what growth looks like. It looks like, in this case, almost like a menorah. You can't miss it if you're actually in the focal depth of the scope. So some of the other carbon in this picture is just either out of the focal depth, is smaller, so it doesn't have as heavy biofilm growth on it, or doesn't have growth. It depends on which ones you're looking at. And you don't necessarily see growth on every piece, especially when you scratch it out of core, but you don't really know what its orientation was. But what I have down in the red circle are pieces of carbon that are the size of colloidal carbon to give you an idea of what that looks like, relatively speaking. Okay, now on the right, again, you see a piece of carbon with lots of growth on it. And you wonder, do the other smaller pieces also have growth? So look at the one that I've circled in yellow. That would be this one right here. Now I've tried to magnify it, and I understand it doesn't look very clear, but that's the limitations of the scope. You will see right here, tree-like structure, biofilm, clear biofilm, or tight, thick biofilm here. So yes, even the smaller pieces in this case are growing. Now I circle these pieces here, look a little bit like the Philippine Islands. Maybe looks a little bit like Australia or something. But that is the picture on the right that I again have magnified. And yes, it doesn't look good, but it's the best I could do. Notice the little island that I said looks like Australia with little horns on it. Notice how it's pointed. You're not going to get much in the way of pointed activated carbon. Most of the activated carbon, at least visually, is going to appear to have a roughly smooth shape. These points are probably also microbial growth. I show you a couple other pictures of microbial growth. The one on the left, I guess I just showed you because I like it. It's pretty. The one on the right shows you both a piece of carbon the size of a larger piece of powdered activated carbon with clear growth on it. And then, of course, the larger piece, heavier growth. It appears that the larger the piece of carbon is, the larger and heavier the growth. I think it's the greater concentrated resource is what it becomes. Now this is an interesting picture, and this is just good old Western Kentucky clay. It's out of a soil core, and what I did is I took a razor blade and cut a really, really thin piece of clay. Then I took a toothpick, stuck it in a colloidal carbon, a major brand name's carbons, spread it around in water, and then very lightly rolled the toothpick up the clay so that you see in the top right-hand corner the clay, and in the left-hand side you see the clay with colloidal carbon on it. So this little black spot that my pointer arrow is pointing at right now, whoop, it disappeared on me. Right there, let me move it right here, pointing right there. That is a piece of colloidal carbon. And all the black you see in this picture is colloidal carbon filling the spaces within the clay. Now, what you'll notice real quickly is that the colloidal carbon fine-grained character of it is pretty much the same as the fine-grained character of clay itself. So how this kind of a material would become habitat for microbial growth, well, I don't understand how that would happen, but nevertheless, here this is, and that's what it looks like. Now, in some cases, you can see that it's lined pits. So in all, to be fair, the colloidal carbon is really, really good about lining things. I mean, it's almost like spray paint. In fact, you can't really get it off once it's on there. This is a close-up. I know it's very shiny and it looks like a handful of jewels or something, but this is a close-up of the picture I just showed you. So here's a pit that's filled with colloidal carbon. It looks almost like caviar filling the pit or something. Here, the little balls here are all colloidal activated carbon. And then you see some other items that you can probably, maybe that's a little piece of quartz or something, right? You got some little green something here, whatever. But again, the granularity is not, well, it's in this case, it's even less than that of the clay as it appears. Now, I did take another picture, that's this one here, trying to get a clearer photograph. And actually, this is my cell phone looking through the lens of the eyepiece of the camera. So again, the little black spots here are colloidal carbon on the clay. And the point is, it's not much as far as habitat goes. It does stick well, though. Now, GAC and PAC, at least as we do it as a company, is in place by high energy injection. And the interesting thing about high energy injection is it actually lifts the geology and inserts the carbon. And that actually gives you not only the space of the carbon, but the space that is made by the process itself. So the picture you're looking at on the right is a 2.25-inch core log split vertically to display the carbon seam. And you not only have the carbon seam, which I'm drawing along now with my mouse pointer, but you also have carbon that is headed toward you. This is space. This is actually habitat. And it's habitat because I showed you earlier. Here's another picture showing actual space for microbes to grow and a seam of carbon in. This actually is central Kentucky clay in this case. So, in order to make colloidal carbon, you have to mill the carbon. And the way that's done is you could use a ball mill or a jet mill, and you drive the carbon down to an average size of 2 micromillimeters. When you do that, you remove those surface features and fractures, which I showed you earlier in this presentation. That's what you have to do to attain that size. By doing that, you no longer have a place for microbes to attach and build habitat. This does not affect the absorption. The absorption onto colloidal carbon is just as good as on GAC and powdered activated carbon. And in some ways, for some uses, the absorption is actually better. But colloidal carbon is too small to act as a resource. We saw even with the larger grains of carbon that I was showing you, that the GAC, the large pieces, appear to be better resources than even the smaller pack. So If you are producing a carbon that is the same average size as the bacteria, you are not providing it a place to live, a place to grow, and a place to build up a population that metabolizes your contaminant long term. So the assumption is that microbes grow on colloidal activated carbon in the same manner in which I showed you on GAC and PAC. This has not been demonstrated. And in what space would they grow when the material itself is the same size? Colloidal activated carbon is too small to encourage adhesion. GAC and PAC, I've demonstrated adhesion and microbial growth. Colloidal activated carbon does not demonstrate biomass or community structure. I have demonstrated both for GAC and PAC. The long-term basis for any sustainable treatment, the treat, is a robust microbial community. And to demonstrate that a product or a process or an item that you're using will clean up a site, you must show that it is robust and sustainable for the long term. Thank you.
Thank you, Ed. That was excellent, as always. We do have a few questions that have popped in here. Let's get to those. Let's see. You said that the high energy injections lift the geology. Can you explain a bit more about how you are making space while adding mass?
Yes. When I say lift the geology, some carbon contractors will use what are called tiltometers. And you can actually lay the tiltometer on the surface of the ground and measure the lift as the carbon goes into the ground. So you're not merely crushing the material around you, you're actually lifting as you insert. So by doing that, kind of like a wedge going into a piece of wood lifts it, by doing that, you're actually making space for the carbon. And then, of course, the carbon itself has space within it for microbial growth.
Okay, and this is very much related. You said if you inject colloidal carbon through low pressure injection, you don't make space for microbial growth. Maybe you could reiterate a few of those points you made on that.
Yeah, if you're doing low flow, low pressure, the goal is usually stated as trying to follow the original pathways that the release followed. Now, in this particular case, you've got these small, very small particles, and they're going to, if you even hit those pathways, they're going to line the pathways, thereby probably constricting them as opposed to expanding them. But beyond that, you are making the assumption that the pathways that are important to cleaning up the contamination, the pathways that are available for you to follow today, are primarily and in majority the same pathways that were active at the time of the release. If this were a release that occurred recently, I would buy that argument. But most of the people listening to this call their big problems are sites that are 20, 30 and more years old and had releases which occurred over many years. During that time, you had what? You had the water table move up and down. You had seasons occur. You had development around the area and infrastructure put in and pulled out. You have vibrations from moving trucks and cars. You have all kinds of things that happen over the years that change the preferential pathways that are available both in the aquifer and in the overburden over time. So the assumption that you're going to just let it kind of flow and it'll follow the pathways that it once flowed, and that those pathways wouldn't be potentially clogged by a very small grained item is a bit too much of an assumption, actually.
Yeah, that's a great point. I'll just real quick, someone pointed out that the microbial growth on the GAC pack reminds them of branch coral on a reef in the ocean, which is, I think, an interesting analogy to what's happening when you put the carbon in the ground and create that platform for growth. There's a question here. Great presentation, Ed. Just wanted to hear your thoughts on the appropriate application of colloidal activated carbon. Most applications seem to benefit from the low grain size, i.e. ease of injection, occupation of smaller pore spaces, enhanced amendment distribution. Do you see these benefits being outweighed by the drawbacks from a bioremediation standpoint? Or is the colloidal activated carbon most useful for recalcitrant compounds where biofilm growth is not the priority?
Yeah, and you know that we've at RPI we've looked at colloidal carbon ourselves and could sell it if we chose to. It's a really interesting and admirable effort to try to make a product that people can install with the equipment that they have. So I don't want to be, Mr. Negative here and unkind or anything like that. The absorption kinetics of colloidal carbon is actually very good. So I hate to bring this up, but I will. If someone said to me, you have to inject carbon product for PFAS, you have to inject it, and all we want to do is slow it down or stop it, then I would probably choose the colloidal carbon for that purpose. The difficulty is if you don't have a treatment mechanism, I would think that ultimately you're just building a hazardous waste landfill that should probably be permitted. But nevertheless, the fast kinetics of absorption and the fact that you were going to try to inject it would probably be the best approach. If you were going to use a permeable reactive barrier, it probably doesn't make too much difference, the grain size in that case. So yeah, that it does absorb, it absorbs well, it really does. But for most processes that we're interested in, we do ultimately want that treatment mechanism. I don't think that really answered the question completely, but that's what comes to mind.
No, I think it raises several good points there. Another one here, does the extension of the bacteria create any increased capacity for biodegradation?
Yeah, biodegradation is directly related to the number of microbes capable of degrading. And so the larger population you have, the assumption is the larger or the greater the capacity to biodegrade. Now, there are some misnomers in that. So for instance, almost everybody knows Dihalocoides McCartae is an example. And, if I said I had a TCE site that had 100% Dihalocoides McCartae, some people would get really excited. But what I would do is get really worried because Dihalocoides McCartae needs acetate. And where does that acetate come from? Other microbes have to make the acetate to feed it. And this is only an example of what is true of most microbial populations, that the population needs other members. For instance, most microbes do not make all the amino acids they need. They have to get it from other members. There are microbes that actually make polyester, and they turn themselves into big polyester bladders. And what happens in the biofilm is they're hung up like hams in the barn. They feed the larger population when times get rough. That goes to sustainability and persistence. So the microbial biofilms in populations can become very complex. And that's what you want. You want that complexity. You want that mixed population. You want something that's able to sustain itself. So yes, the extensions are visual representations of populations that have the diversity and robustness to last.
Yeah, and I think that really crosses over into this other question where it was regarding the biofilm. Why does the population need to be made up of anything other than key degraders?
Yeah, and I think I touched on that with the Helicoides macarte example. And that is, most degraders are kind of specialist. If you look at the biofilm as a city, you know, you don't want a city that's just filled with doctors and lawyers, right? Somebody has to fix the plumbing. Somebody has to run the grocery store. And I'm being a little silly, but the same thing is true in biofilms. The specialists oftentimes are needy and need help from other microbes.
And do you have data, have you collected data that the microbes that you're seeing on the carbon are those microbes that are capable of degrading?
Yes, I've sequenced what was in the seams that I've shown you in this video, in this presentation. But to go over that is really a whole different presentation. And what you do see are populations that are richer and have a more diversity of metabolic processes than in the controls.
So if I'm understanding correctly, you're saying that the population is changing when it grows on the carbon.
Oh, absolutely. Yeah.
Okay. Let's see, when injecting electron donor with colloidal carbon, does the donor substrate or volatile fatty acids sorb to the colloidal carbon? And would these VFAs adsorbed to carbon still be available to the microbes? We'll start with that. They add on after that. I'll let you address that.
Yeah. Okay. So when you mill the carbon, you basically end up with your micropores to the outside. So yes, you could bind some of those volatile fatty acids onto the micropores. Are they available? The answer, just as with most things that bind into the micropore structure, the answer is both yes and no. They're bound pretty tightly, so they're not necessarily immediately available. But certainly if you were to build up, which you're not with the microbial, with the colloidal carbon, but if you did build up biofilms, the biofilms can manipulate the concentrations around the carbon. And you can also, exoenzymes from the bacteria can help remove things from the carbon. So yeah, I wouldn't poo-poo the idea completely that the fatty acids that went in with a colloidal carbon are available. And the ions, of course, that you put in are charged. So the activated carbon is not particularly interested in those ions. So yes, they will also be available. And that's one of the reasons why some of the pictures I showed you, when I was showing you some of the colloidal carbon, I was showing you carbon that was a product from another company. And in some cases, I was showing you colloidal carbon that was injected in with our products so that the electron acceptors and everything else that was present with the GAC and the PAC was also present with the colloidal. Because otherwise, it just wouldn't be a fair comparison, would it?
Sure. And I think you've addressed the second part of this question, which was, could the VFAs also compete with sorption sites on the carbon? And it sounds like in general, you're saying no.
No, I would say that they could, yes.
Oh, okay.
Yes.
My misunderstanding. Yeah. Oh, okay.
I probably wasn't clear, and I apologize if I wasn't.
Do you have a slide or data to show grain size of your carbon products for GAC or PAC? Are there preferences for grain size to type of contaminant?
I think our looking at this and becoming aware of the carbon as a platform for biological growth is making us consider that topic. Not only am I saying that I think the colloidal carbon is not the right size grain size, we may be using that information ourselves to answer the question of what is the right grain size. At what point, what is optimal for microbial growth relative to, being able to actually get it in the ground and things like that. So, yeah, there are differences. Now, as far as contaminants go, yes, you will see that some contaminants, and I should be able to think of a one right now, but nothing's coming immediately to mind. Some contaminants will absorb a lot better when you have carbon that has mesopore structure, that middle size pore structure, which is from 2 to 50 nanometers. And they need that. So for instance, if you had a coconut carbon, which has a lot of mesopore structure, it does actually absorb some things better than coal-based activated carbon, which has less mesopore structure. So yeah, the pore structure can certainly change with the kind of carbon, and the pore structure changes with the grind of the carbon. So yeah, there's some optimization to be done there. I just don't know exactly what it is.
And my understanding is that all of the activated carbon products on the market are coal-based for their use for in situ remediation, correct?
Yes, I think everybody's a big statement, isn't it? So let's not say that. Let's go. Yeah, no, I didn't. I said it myself first. I think the major players all use coal-based carbon.
Okay.
Okay, so there are probably other people out there that use coconut-based or whatever, but you do get different results.
Okay. Interesting question here. What percent of benzene in groundwater gets sorbed, and what percent treated by bio?
Yeah, Ed, I'm sorry, I really couldn't answer that because there are too many other factors to consider. So it's an equation with too many unknown inputs. But as a general statement, it depends on how much carbon you put in the ground, how much gets absorbed. There is a limit to how much you can get to the point where you put more carbon in the ground than actually does you any good. So you don't actually have to fill up all the pore volume of the subsurface in order to max out the efficiency of carbon. And then, of course, you have to know, what the concentration of the benzene would be to come up with percentages. And then ultimately, you want to have a sense of, what point in time you're actually answering the question. So initially, what you see is the carbon absorption of benzene is really fast. You'll see benzene concentrations maybe sometimes drop to 0 across your wells. And you'll think, woohoo, I've made it. But remember that benzene is the most water soluble, if we're assuming we're talking about gasoline here, the most water soluble of the components. And it'll oftentimes win the foot race to the carbon. But when other components, such as your xylenes, show up, which actually have a higher heat of absorption, they'll kick the benzene off the carbon. So then you will see a kind of a rebound of the benzene. Now that freaks people out, but it usually doesn't come to rebound anywhere close to where it was prior to injection. And then as the microbes begin to biodegrade the components of the gasoline, then you see the benzene levels start to drop down again as the carbon is cleared or as they are degraded by the microbes. So that's a long answer to cover up the fact that I can't answer the question. It was a good one, though.
Well, and again, I would refer everyone to the trapandtreat.com website for RPI, also the ASTEnviro.com website, where both websites have a nice library of recorded webinars that go into pretty good depth on a lot of these different concepts that are coming up in these questions. So, and you can always reach out directly to Dr. Winter, reach out to me, reach out to any of your contacts that you have in our companies to talk about these things further too. But that's the last of the questions that we've had pop in, Ed. So I want to thank everybody again for your time today and for all of your excellent questions. And you know, I'm hoping that we hear from you with, you know, if you have any specific sites you'd like us to screen for the applicability of these technologies. If you'd like to do any kind of data review and see if there's any data gaps we can help you fill, then that's certainly something that we specialize in as well in order to figure out the best technology for your site. So thanks, folks. Hoping that you have a great rest of your day, and we look forward to working with all of you at some point in the future.
Okay, well, I appreciate everybody listening. Have a good day.